today's video is gene editing, and how gene editing can be used to fix errors in the human genome, which in turn can cure diseases or help create treatments for things such as cancer and HIV. However, to understand how the CRISPR-Cas9 system achieves this, we must first take a look at genetics and DNA. So let's talk about genetics. Genetics is a scientific study of heredity, or the passing down of traits. So a gene is the basic unit of heredity, and it carries information that determines not only your physical traits, but also other factors such as your susceptibility to diseases. So what makes a gene then? A gene is comprised of DNA, or deoxyribonucleic acid, which is found in the nucleus of all living cells. DNA is a double-stranded structure with a sugar phosphate backbone and nitrogenous bases. There are 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. A pairs with T and C pairs with G. So once these pairs are matched up, it then coils to form a double helix structure. So now let's talk about genomic mutations. Mutations are changes in the DNA sequence of an organism. They are caused by either environmental factors or during cell development. There are 2 main types of mutations: point or gene mutations and chromosomal mutations. So these have varying effects. Some are the cause of diseases, whereas others have no effects at all. Mutations found in gametic cells may be passed down to offspring. Finally, to the point of this video, how could gene editing be used to fix these mutations? So CRISPR-Cas9 is a recent technological advancement that allows scientists to precisely edit the human genome by adding, removing, or modifying genetic material. So the CRISPR-Cas9 system has 2 main parts: the Cas9 enzyme and the guide RNA. The Cas9 enzyme is a type of endonuclease that acts as a molecular scissors, cutting the DNA at a spot specified by the guide RNA. The guide RNA is a type of ribonucleic acid, which is bonded to the Cas9 enzyme. So a variety of edits can be made with this technology. Disruption is when the Cas9 enzyme makes a singular cut with one guide RNA. The 2 ends are then repaired through natural processes, which can lead to gene inactivation. Next is deletion, in which 2 separate cuts are made. Those 2 ends are joined after that middle piece is removed. Finally, we have a correction or insertion, in which the genetic material is cut. A new template strand is brought in, and then this can correct a gene or create a new gene. All of these processes can be done ex vivo, outside the body, or in vivo, inside the body. Gene editing, and CRISPR in particular, are still in their earliest stages of understanding, from curing genetic disorders to creating genetically modified crops, and even creating treatments for HIV and cancer. The opportunities with this new technology are boundless.